Hey, we're back for what's probably not the last, but getting near the last lecture on the urinary system. Probably gotta do at least two more, hopefully. So I shouldn't waste time with this little preamble here. Let's get started. Last time we were talking about reabsorption in the PCT. Reabsorption in the PCT. So check it out. Here's our glomerulus. We got some glucose in the afferent arteriole. Dun 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 dun. Glucose comes in the glomerulus. Glucose gets filtered. Not all of it. Some of it goes in the afferent arteriole. The glucose that gets filtered, as it goes through the PCT, 100% of it gets reabsorbed. All right, that's the normal situation, meaning no glucose is in the P. However, what if there was too much glucose in the blood? If there was too much glucose in the blood, then there's going to be too much glucose in the filtrate. And we're going to we're going to saturate the receptors for glucose, the proteins that shuttle glucose out of the PCT. And when that happens, when those proteins are saturated, they'll move as much glucose as they can, but then there'll be some left over that gets peed away. We'll be reabsorbing glucose at our transport maximum, but we can't get it all, and so it gets peed away. All right, this is what happens in people. I just said in peepees. Freudian slip there. In people who have diabetes, untreated diabetes. And it's, one of the things about reabsorption is you can only reabsorb so much. If you've got too much to reabsorb, you physically can't do it. All right. On that note, let's talk a little bit about reabsorption in the good old distal convoluted tubule. There it is. The reabsorption here is hormone dependent, all right? It's not obligatory. You don't have to do it. This is where you fine tune the contents of the filtrate in order to maintain homeostasis of the blood. And hormones act on the DCT to up or downgrade the amount of reabsorption that takes place there. Two hormones specifically that act on the DCT include aldosterone, who we'll talk about in a moment, and good old parathyroid hormone that I know you remember fondly from A&P1. It is important in calcium balance. Parathyroid hormone increases your blood calcium levels. One way it does that is by increasing the reabsorption of calcium in the DCT. And by reabsorbing more calcium, we pee less away and bring blood calcium back up. All right, the loop Henle. The descending limb and the ascending limb actually reabsorb different things. More water reabsorbed in the descending limb, more salt reabsorbed in the ascending limb. And what that is going to do is help build a concentration gradient right here, where my cursor is making circles. And the concentration gradient right here is going to help us ultimately concentrate our urine, make the urine darker, steal water from it. We'll talk more about that later as well. I keep saying we'll talk more about things later, and we will. I know you're getting excited about this, and then I say we'll talk about it later. I apologize for that. Collecting duct. Last part where we do some reabsorption. There's the collecting duct. We have urine in the collecting duct, urine from several different distal convoluted tubules. And the thing we reabsorb in the collecting duct is water. Depends really on two things. Hydration levels in the body, which is going to determine how much antidiuretic hormone is being released. Antidiuretic hormone causes you to, it doesn't cause you to, it sets the stage for reabsorbing water in your collecting duct. The more ADH you have, the more water you reabsorb from the collecting duct, and the less pee you end up peeing. All right, let's talk briefly about secretion. Secretion is the opposite of reabsorption. Realize that as blood comes into the glomerulus, we cannot filter everything. All the bad stuff doesn't go this way, like that. All right, all the bad stuff doesn't get filtered. Some of the bad stuff stays in the blood, in the efferent arteriole. So that bad stuff, and by bad stuff I can mean wastes, things like urea, uric acid, creatinine, certain ions. They end up in the peritubular capillary, where they're then actively moved from the blood into the filtrate. And if you take a real physiology class down the road, you might get into the mechanisms of how that occurs. But suffice it to say, that's it for secretion for you and me right now, and for your upcoming exam. All right, now we get to revisit some things. We get to revisit the loop of Henle, the collecting duct, ADH, and we get to talk about regulating the concentration of blood. One way to regulate, or another way to talk about blood concentration is the blood osmolality, which is basically the number of particles we have in a kilogram of plasma. And we got to maintain it. I mean, if it's too low, water will move into our cells, swelling them. If it's too high, then water will be stolen from our cells, shrinking them. And cellular swelling, cellular shrinkage can affect cellular performance. We don't want that. So we actually measure blood concentration in our hypothalamus. There'll be cells called osmoreceptors up there that measure the osmolality of blood. And then we're going to adjust blood concentration by regulating how much ADH, antidiuretic hormone, is released from the posterior pituitary gland. Let's walk through let's not walk through a scenario. I forgot I got another slide here. Just a reminder that ADH increases the reabsorption of water from the collecting duct. All right, now let's walk through a scenario. Suppose blood osmolality was high. This means blood is too concentrated, which means we got to get some more water into it. Let's see how that happens. Well, the high concentration is going to be sensed by osmoreceptors, specialized neurons in the hypothalamus. They're going to tell the posterior pituitary gland to increase the release of ADH. ADH is going to increase the permeability of the collecting duct to water, allowing more water to be stolen from urine. As water is stolen from urine, it goes back into the blood, and thus blood osmolality falls. This type of reabsorption of water is called facultative. We do it when necessary, as opposed to the obligatory reabsorption we did in the PCT. All right, super. You should, of course, be able to figure out what happens when blood osmolality is too low. And we've got to answer 5 questions here. You got to tell me what ADH will do to all 5 of these. And right here we've got an MRI of a girl with a tumor called a craniopharyngioma. This girl gets crazy thirsty, always has to pee. You got to tell me what brain structure is not working properly. All right. So we mentioned that ADH makes the collecting duct more permeable to water. Oops, and I just lost my microphone off the top of my head. Note to self, don't lean down, don't lean forward and try and pick something up off the floor while wearing headphones and a microphone because it can fall off. Let's see. What else? What else is necessary for water reabsorption to occur? I know that's what you were wondering. Well, there's got to be a gradient, all right? There's got to be a medullary osmotic gradient. Remember the good old loop of Henle? Loop of Henle reabsorbs water, reabsorbs salt. And what that does is it builds high concentration in the interstitial fluid in the medulla of the kidney. So you actually get more and more concentrated the lower you go in the medulla of the kidney. And so, if you notice here in the collecting duct, urine's kind of dilute here, but water is constantly leaving and getting picked up by the blood in the vasa recta. Water's able to leave and go through here because of ADH. But the force, the osmotic pressure pulling the water through is because of this high concentration out here, and this high concentration was built by the loop of Henle. All right, let's see. Still got about 40 seconds to go. Let's do a little more. What would happen if ADH wasn't present? If ADH wasn't present, well, the osmotic gradient would still exist. However, without ADH, there's no permeability, and the water stays in the collecting duct, stays in the pee, and we pee away dilute urine. We talked about the kangaroo rat in lab and in lecture. Make sure you understand why he has long loops of Henle. And next time, we'll talk about aldosterone. See you later.